Hello everyone. In this talk, I present a global view of upper mantle stratification using a new method called CRISPR-F, a technique that removes echoes and noise using sparse radon transforms. The talk is in three parts. First, I review existing observations and models of upper mantle stratification. Second, I introduce a filter that can remove reverberations buried in converted waves. And third, I describe new constraints and the new taxonomy of upper mantle stratification across the United States. In this slide, we see global observations of upper mantle layering that have inspired three major explanatory models. On the left is the model of melting to explain the top and bottom boundary of oceanic low velocity layers. On the top right is the metasomatism model, and below is the attenuation model, both of which explains discontinuities within the stable and old lithosphere. As the search continues for a representative model to explain upper mantle stratification, a new controversy has surfaced on how robust the observations are. The issue in question is this signal processing step called deconvolution. Some works show that improper deconvolutions can introduce side lobe artifact results. On the left, you see an image of upper mantle stratification with and without deconvolution. These differences suggest that many detections of the mid-lithospheric discontinuity are not robust, inspiring a re-evaluation of global observations. We show on the left that most authors prefer to use S-wave instead of P waves because they are not overprinted by reverberations generated in the crust. This is an advantage. However, because of the low resolution coupled with deconvolution side lobes, S waves are not ideal. P waves, on the other hand, are higher resolution and are overprinted by reverberation as seen on the right. Our goal is to remove these reverberations and make P waves useful for revealing fine details in upper mantle structure. How are we going to do that? The method is CRISPR-F. And the central idea is radon filters applied to P2S receiver functions. Here we show how the method works. CRISPR-F is an acronym for clean receiver function images obtained with sparse radon filters. On the top is a standard single station P2S receiver function stack obtained without CRISPR-F. Below is the result after denoising with CRISPR-F. CRISPR-F has denoised the P2S receiver functions, making it easier to interpret. Step 1 transforms the P2S receiver function data D into a sparse radon model M. The direct conversions which show up on the top region of M and are indicated by open circles and are separated from multiples in the bottom region identified as squares. The second step applies a filter to isolate these direct conversions before step 3 transform the model back into the data domain as a denoised P2S receiver function. With this new tool, we revisit decade-old observations from a stable continental regions of North America, Southern Africa, and Australia. We show on the top right our station distribution and below single station P2S receiver function stacks across the US. As you can see that without CRISPR-F, it is very hard to identify upper mantle structure. But with CRISPR-F and machine learning, we will provide compelling results on upper mantle structure. A machine learning algorithm called sequencer is applied after CRISPR-F to convey the improved information content. Here, you see the sequencer algorithm in action. The image of Einstein on the left is shuffled into a random sequence in the middle. Applying the sequencer algorithm, reconstruct the original image of Einstein. And we do the same for our receiver function stacks. On the left is our P2S receiver functions before CRISPR-F denoising, and right is after CRISPR-F denoising. The experiment on bottom left shows that without CRISPR-F denoising, the sequencer fails, as you cannot see any pattern here. But after denoising, information content is improved and entropy is reduced, and a clear pattern of upper mantle stratifications become visible. The velocity reductions on the top and velocity reductions on the bottom implies that. Further classification using hierarchical clustering further refines depth-dependent and polarity-dependent stratification as shown in the top part and the bottom part of this slide. A similar analysis has been applied to Africa and Australia. Here we show that sharp mid-lithospheric discontinuity associated with a velocity drop is indeed most prevalent discontinuity, and it is indeed global. We also point out that we detect more discontinuities across and below the lithosphere, although this class is less prevalent. Stay tuned for more results, and please visit posters from John Joel Legray and Steve Carr in the following EGU sessions. Our result in the US shows that Mantle is stratified. We introduce a taxonomy of this stratification underneath stable continents. Single discontinuities are marked in circles. Paired discontinuities are marked in squares. Intralithospheric discontinuities are in green, transitional are in yellow, and sublithosphere are in pink. Based on this new taxonomy, we suggest that the single discontinuities are better explained by the subsolidius attenuation model, while the others fit more alternative models. A robust test of our constraints will follow when global results are finalized. I'll stop here and leave up the slide of the team that has worked on this project. We have reviewed upper mantle stratification in 3 major continents, showing the fine details of upper mantle structure with important implications for evaluating causal models. I thank you all and leave the slide for open questions.